Continuation from the videos that we had to look at, that the effect of enzyme activity, specifically the video was about the effect of enzyme catalase on the effect, or it was the effect of enzyme catalase on, we are looking at the effect from the experiment or from the video that we had to look at, effect of enzyme catalase on the composition of hydrogen peroxide. We're able to view that when enzyme catalase is added to a solution of hydrogen peroxide, we have effervescence of colorless bubbles, and those colorless bubbles were coming from the composition of hydrogen peroxide, which gets broken down into water and oxygen gas. So we are looking at enzyme catalase breaking down hydrogen peroxide to oxygen and hydrogen gas. Now, we had experiments under different conditions. In one of the conditions, there was added, or they added an acidic media. They got the tissue of the liver and it was added to an acid. In another one, it was added to an alkali. And in another one, the tissue was boiled. In another one, the tissue was added in its real size. So we look at different factors affecting the rate of enzyme controlled reactions. And one of these factors, we have temperature. Another one, we have concentration of the substrate. We have pH of the medium, presence of activators, and presence of inhibitors, plus the concentration of the enzyme. So these factors affect enzyme activity differently. For example, factor number one, we are going to look at concentration of a substrate. We are calling a substrate a substance acted upon by the enzyme to form simpler products. So a substance that is being acted on by the enzyme is what we call a substrate. In other words, in the experiment that we looked at, we are looking at hydrogen peroxide being a substrate. It is the one that is being acted upon by the enzyme catalase that is present in liver, as we can as well find it in Irish potato cubes. When you get an Irish potato, you can cut it into small pieces, and instead of adding in a liver, you can add in an Irish potato. It also has an active substance known as enzyme catalase. So what happens if you increase the concentration of the substrate, then you also increase the rate of reaction. So we are looking at, if we increase the substrate concentration, the rate of enzyme controlled reaction also increases, because there will be much substance that will give more products. So the rate of reaction will increase, so as that the enzyme can work on those substrates that are present in high concentrations and give you more products. And if the substrate concentrations are low, then the enzymes also work at a slower rate. However, when you continue to increase the substrate concentration, you will have no continuous increase in enzyme controlled reaction, or the rate will not continue to increase. Why does the rate not continue to increase? The rate will not continue to increase since all the active sites on the enzyme are fully saturated with that substrate. Now, a point reaches where the continuous increase in substrate concentration, the rate of reaction becomes constant. It becomes constant because the enzymes that are present to combine with these substrates are fully saturated. On the enzyme, we have a specific part, we have a specific position, which we call the active site. it is this active site which combines with the substrate. It always has a shape that is complementary, the shape that is similar to the substrate. Now, a point reaches when the continuous increase in substrate concentration does not bring about a continuous increase in enzyme controlled reaction or in the rate of reaction. So when you look at this graph, we are having an increase in substrate concentration and the rate of reaction increases rapidly. But where we have the constant rate, we are having the rate of reaction, now it will be limited by less enzymes that are available to combine with the substrates. So that means that when you get a solution of hydrogen peroxide, you add in the liver tissue, you will have effervescence of colorless gas coming up. But the more you increase the enzyme or the more you add in more tissues, you will bring more reaction, you will have more products coming out, meaning that when you had not added more liver tissues, the rate of reaction was limited by less enzymes that are present in the liver tissue. So first, where we're having an increase, the rate of reaction is being limited by substrate concentration. In the middle, where we have a constant phase, we have the rate of reaction limited by less enzymes. But if more enzymes are added, the rate of reaction increases. So it is like a combined effect of two factors. Rate of reaction increases with increase in substrate concentration, but the rate of reaction also increases with increase in enzyme concentration. So a decrease in enzyme concentration also lowers the rate of reaction, and an increase in enzyme concentration also increases the rate of reaction. We have the second factor, which is temperature. Now, we are saying that enzymes work best at optimum temperatures of approximately 35-37 degrees Celsius in humans. Now, what happens is when the temperatures are very low, we have the rate of reaction being very slow. This is because enzymes need to be activated, and at low temperatures, enzymes are less active, so they are still inactive. But as the temperature increases, the rate of reaction also increases gradually until it attains a peak. The rate of enzyme-controlled reaction will increase with increase in temperature until it attains the peak. And at that temperature where it has attained a peak, or where we have maximum rate of reaction, we say we have reached the optimum temperature. At this temperature, enzymes are fully activated. They are combining with substrates. They are fully activated for combining with the substrates. Hence, we shall have very many products coming up. So the rate of reaction will be very fast and high at that point. But if you continue to increase the temperature, we have the rate of reaction again decreasing. This is because these enzymes are protein in nature. Being protein in nature, they lower or their structures can be altered or changed or completely destroyed in presence of extreme heat. That means the temperature that is beyond the optimum denatures or changes completely the structure of the enzymes. Denaturing means they are changing the structures. The structure is being destroyed. The original nature is being distorted. That is what we are talking about. So at extremely high temperatures, we have the rates of reaction reducing because the enzymes are being destroyed. Now, when the enzymes are destroyed, the active sites, those active binding sites that will be combining with the substrates, they will be changing. And eventually the substrate, which comes with a complementary shape that wants to bind with the enzyme, it will find that the enzyme has completely changed, and as a result, there will be no reaction taking place. So at temperatures beyond the optimum or at extreme temperatures, we have the rate of reaction decreasing because there is denaturation of the active sites of the enzyme. As we said, when we are looking at the substrate concentration, when the concentration of enzyme increases, the rate of reaction also increases until all the substrates are being acted upon, when the rate finally becomes constant as long as no other factors are limiting the reaction. So it holds when pH, pressure, and temperature are constant. That means a reaction must be having the favorable pH at which it is reacting. The reaction must be having the favorable pressures, and the temperature should be optimal. And the substrates should be present in large concentrations. So once you have all these factors held constant, once they are favorable for such a reaction, and you decide to increase the enzyme concentration, the rate of reaction will be increasing. These are displayed experimentally on these graphs. The curve shows that there is an increase in rate of reaction as the enzyme concentration is increasing along the horizontal scale and the rate of reaction is on the vertical scale. Now, these enzymes work under specific conditions. And one of the conditions that these enzymes must work upon or must work in, we call it pH of the medium. Now, most enzymes, they are effective over a narrow pH range. We are looking at pH, the potential of hydrogen ions. The pH, we always have acidic, we have alkaline pH, but that is the general. But within that scale, we have the different ranges. Acidic ranges from 1 to 7, and at 7 we have neutral pH, and alkaline ranges from 7 to 14. Now, from 1 to 3, that is strongly acidic. Then from 3 to 7, that is weakly acidic because it is coming to neutral pH. Then from 8 to 9.5, that is weakly alkaline, and from 9.5 to around 14, that is strongly alkaline. Now, these enzymes, they do not work at every pH. They have their specific pH ranges where they work from. Particularly, you look at enzyme pepsin in the human stomach. It can have its maximum activity. You are seeing the rate of reaction on the vertical scale, and down is the pH. So you see pepsin where it has the peak, that is the pH. The pH that is varying with on the horizontal scale is that point where it has maximum activity. That means pepsin enzyme in the stomach has its maximum activity, or it acts upon the proteins. We shall look at that under digestion. We shall find out that this pepsin is breaking down the proteins into the peptides. So it completely breaks down the proteins very fast into peptides at a pH between 1.5 to 2.5. And within the stomach, the juice that is produced by the gastric glands, known as gastric juice, consists of hydrochloric acid, which is responsible for this acidity media within the stomach. You look at enzymes within the duodenum, particularly trypsin. It is working at a pH range between 8 to 14, but its maximum activity is between 10 to 12. That is around 11. You also look at amylase. Its maximum activity is around 7. That means amylase works best around the neutral pH. around there, 7, 6.8, around that. So if anything happens and you have a reaction where you are breaking down proteins into peptides by pepsin, and you change the pH by addition of an alkali, you will lower the rate of reaction. Sorry. Because the pH will have been changed, it will distort the working action of the enzymes. The enzyme's nature will be disturbed, so it will not have favorable conditions to combine with the substrates, hence giving you products. So enzymes work best over a narrow range of pH, and any sudden change from the narrow range of the pH of such an enzyme, it will affect the rate of reaction by lowering it. That is the pH of the medium. Now we have other enzyme factors that affect the rate of enzyme controlled reactions. And we are calling these guys inhibitors. Now, something that favors or something that is supporting a process is called an activator. We are looking at activators increasing the rates of reactions. But if these activators are not present, the rate of reaction will be affected and lowered. Now, instead of providing an activator, you bring in an inhibitor. This inhibitor, we are saying we have two types of enzyme inhibitors. One is competitive and another one is non-competitive inhibitors. Now, under competitive, we say that these inhibitors, they possess the similar structure to the substrates. So now, instead of the enzymes combining with the substrates to give us products, if an inhibitor is present, the enzyme will combine with the inhibitors, giving us no reaction taking place, because they will have to reduce the enzyme concentration. Now, most of these inhibitors, they combine with the enzymes completely. So the enzyme concentration will be reduced. Hence, presence of enzyme inhibitors decreases the rate of reaction because they interfere with enzymes completely destroying their action. That is competitive. But the non-competitive inhibitors, for them, they come and attach somewhere on the enzyme. Now, their presence destroys the active site. It distorts the nature of the active site. After distorting the nature of the active site, that means that the active site has been distorted. Hence, the enzymes are destroyed. That means that the substrates will not be able to combine with the enzymes that have been destroyed. So whether the inhibitor is competitive or non-competitive, it lowers the rate of reaction. Particularly, you normally listen to people, they say these guys have died, they were cooking from inside the house. Now, we have the enzyme that is specified, or we have a protein that is specified for transporting oxygen in blood, and that is hemoglobin. This hemoglobin is present in red blood cells. It transports oxygen from blood to the tissues where oxygen is required for respiration. Now, inside the house, when you are burning, you are using charcoal, or as you are cooking. This charcoal contains carbon. Now, carbon reacts with oxygen in presence of excess oxygen to give us a gas known as carbon dioxide. And if the oxygen is limited, it gives us carbon monoxide gas. Now look, hemoglobin has a high affinity for oxygen, but it has a more affinity for carbon monoxide gas if it is present. So that means as you are burning from inside the house and it is closed, there is limited oxygen present in such a house. So what happens is you have incomplete combustion of carbon. Carbon reacting with limited oxygen, giving you carbon monoxide gas. Now, this carbon monoxide gas will go and combine with hemoglobin. Instead of hemoglobin combining with oxygen to give you oxyhemoglobin, hemoglobin will combine with carbon monoxide to give you what we call carboxyhemoglobin, which is a permanent substance that is formed. Now look, hemoglobin transports oxygen as oxyhemoglobin. When it reaches the respiring cells, it releases the oxygen and it comes back to pick another oxygen. But if carbon monoxide combines with hemoglobin, it forms a permanent compound known as carboxyhemoglobin. And this carboxyhemoglobin is using up the hemoglobin. Hence, there will be no hemoglobin to pick the oxygen and take it to the respiring tissues. As a result, you suffocate. You are in a condition known as anoxia, where you have limited oxygen present for the body. That is why you are advised not to cook from inside the house where you are staying or where you are living from, because there is always limited oxygen present. So something like that comes up and you hear that people died from the house and so on from cooking. That means carbon monoxide is an inhibitor of a protein hemoglobin. So this guy was supposed to transport oxygen, and if oxygen was present in excess amounts, then we would have rate of combining with oxygen to take it to the respiring tissues increased rapidly. But because it is in limited amounts and the inhibitor is now the one that is present in large amounts, so it lowers the rate of reaction. So presence of inhibitors lowers the rates of enzyme-controlled reactions. Now we said, these enzymes are biological catalysts that help to speed up the rate of chemical or of biological reactions. But these catalysts are categorized into two. We have these biological ones and we have the inorganic catalysts that we normally use in most of the chemistry reactions, in most of the industries and in most of the laboratories and so on. So when you look at these enzymes and the inorganic catalysts, they are not the same. For enzymes, for them we said they are specific in their action. That is, a specific enzyme works on a specific substrate. You cannot get an enzyme that works on two substrates. For example, pepsin, if it is made to act on proteins and convert them to peptides, then it will never react on carbohydrates. So they are so specific in their action. The mode of action of enzymes is explained by a lock and key hypothesis. You look at a lock, it has a specific key. You can't get any random key and you bring it to open a lock. So they are so specific in their action. But these inorganic catalysts, they are not specific in their action. They act on any substrate that is present. The reactions that are catalyzed by enzymes are reversible, but the ones of inorganic catalysts are not reversible. That is, if you can catalyze proteins to peptides and amino acids, you can as well combine very many amino acid units and they give you back to proteins. So the enzymes are protein in nature, whereas the inorganic catalysts are not protein in nature. We look at the enzymes being denatured by high temperatures, and we look at the inorganic catalysts not affected by high temperatures. We look at the enzymes affected by pH changes, whereas the inorganic catalysts are not affected by the pH changes. We look at the enzymes being secreted in living cells and the inorganic catalysts produced in industries. That is why when you got a liver tissue and you boiled it, there was no effect on hydrogen peroxide. Because you will have killed the tissue, you will have killed the active substance that is present within those liver cells and that is present within those Irish potato cubes. So enzymes work within a specific pH range, work at optimum temperatures, in presence of activators, with increase in substrate concentration and enzyme concentration. These are the factors that affect the rate of enzyme-controlled reactions.